All right. So what do you actually need to know about what causes mutations? You should be aware that most mutations occur during DNA replication, and that they can be caused by problems in replication, but they can also be induced by chemicals and radiation. You should be aware that different types of mutagens cause specific types of mutations. But you don't have to necessarily know which one causes what. So for example, mutagen A might cause C to T transitions preferentially, whereas a different mutagen or radiation causes T to T dimers. Because different mutagens and different radiations have different effects on DNA, the way that they're avoided is also different. So some individuals will be resistant to some types of mutations and not others. And this often has to do with repair pathways. Okay. It's important to note too that most DNA damage does not result in mutation. Because DNA repair pathways are actually really good at their jobs. So even though we've talked about this idea that DNA makes mistakes with wobbles and dimers and changes to nucleotide bases, it frequently catches these mistakes through three different major processes. So these are the most common types of DNA repair. I would like for you to know what they are and when you would use one. So let's talk about them a little bit more. So we have base excision repair, mismatch repair, and nucleotide excision repair. Base excision is relatively straightforward. For example, when deamination converts the cytosine base into a uracil, this creates a small mismatch in the surface of the DNA that can be detected. The uracil is then detected and removed, and the baseless nucleotide is removed. This leaves a small hole in the DNA backbone. So this is the base excision part. Base excision occurs when a single base is removed and replaced. The repair system then reads the template on the opposite side and adds the appropriate nucleotide as a match, replacing the C that had been deaminated with a new C. In mismatch repair, there is a larger issue. The DNA repair mechanism does not recognize this G as whatever it should have been in the first place, but it does recognize that there is a problem with pairing here that creates this little bump. Because it's not sure what this G should be, the DNA strand is cut and the nucleotide that is incorrect and all of its neighbors are removed. Then the patch is replaced using the correct template to put in all of the things that should match, allowing you to fix the one that was incorrect. So in mismatch repair, this occurs when it is not sure whether or not this is a result of slipping or incorrect matching. To make up for that, it removes a larger region of the DNA and uses the intact strand as a template to repair the gap. Nucleotide excision repair is specific to dimers. This is because the dimer creates a large where it is unclear how everything should be fitting. Expected, the surrounding DNA is opened to form a large bubble. The entire area is removed, and it is replaced with undamaged DNA. Your book goes into a lot of detail about the enzymes specifically required for each of these three types of repair, but I do not care if you know them or not. What I would like for you to know is that there are three major pathways, that the pathways are different depending on what is wrong, and the basics of what happens. In all the cases, they need to identify a problem, remove either the base or the surrounding region, and then repair using the other strand as a template. Mutations in the genes required for DNA replication repair or detecting any of these often result in increase in cancer rates. So for example, a lot of the mutations that people have that give them a familial chance of having cancer are involved in DNA repair or checkpoint pathways. And that's why they're more likely to end up with mutations and therefore eventually cancer. But these are very effective. So here's the same thing in one little place. Base excision detects damaged DNA bases. Glycosylase is going to snip that little guy out and then repair it. In mismatch repair, we're going to notice that there is one pair that does not match. We're not sure why, so we take out the whole region and repair. When there is a dimer, same thing. We're going to remove a big region because we're not sure where these two T's are supposed to go, and then repair. It also lists the types of things that can cause those. So base excision can result from spontaneous damage. Mismatch repair usually happens from DNA copy errors, so this is the most used type of repair. And nucleotide excision is specific to damage from chemicals and radiation. There are a number of other ways that your body can repair mutations as well. So for example, we can directly change things that are going on incorrectly, but we also have the advantage of using homologous recombination later to heal double-strand breaks and non-homologous end joining to heal them as well. And we'll go into this a lot more when we discuss meiosis. So you don't need to know about them now. Unrepaired mutations, while rare, are not as rare as you might think they are. For example, most people have quite a few of them. In fact, by the time you're age 15, you should have somewhere between 1,000 and 10,000 mutations per replicating somatic cell. That's a lot. And by the time you're 60, you're going to have somewhere between 4,000 and 40,000 in a single cell. So this is why, as you age, you're much more likely to develop progressive diseases, things like cancer or heart disease, because the cells in your body have accumulated somatic mutations over time. One of the things that's interesting to note, though, is that germline cells accumulate mutations at a much lower rate. But that doesn't mean that they don't accumulate them. And in fact, the fact that they do accumulate them is part of why we see increases in birth defects. When mothers are above the age of 40 and males are above the age of 60. Because even the germline eventually succumbs to this process. Now, most mutations do nothing. They're going to occur in locations in DNA where there either isn't a protein, or it's part of an intron that's not necessarily important, or it happens in a cell that's not using that part of the DNA. Most of these will have no effect, but some of them might. There are multiple diseases associated specifically with DNA repair mechanism issues. Most of these have to do with um, being able to prevent yourself from getting cancer. So a lot of premature aging or premature cancer, young cancer inherited traits are coming from this. So for example, xeroderma pigmentosa results in this production of lots and lots and lots and lots of freckles. Individuals who have this tend to have a lot of mutations occur in their skin. They're much more likely to get skin cancer. They're much more likely to have sensitivity or get sunburned because they can't repair their cells as easily. And they also are going to be very sensitive to other things like chemicals that impact their skin. Fanconi anemia is one that's frequently talked about that also causes skin issues, but it also causes issues for the skeleton, heart, and kidneys, and it gets a lot worse as you get older, and usually results in leukemia somewhere around 50 years old, sometimes sooner. So a lot of these progressive disorders that result in an increased risk for cancer are caused by differences in the ability to repair these types of mutations. It's also important to remember that we've talked about a lot of different types of mutations and that these mutations can all cause the same types of phenotypes. So for example, cystic fibrosis is a relatively common inherited disease in the United States. 1 in 35 individuals is a carrier for this disease, and it's higher if you're white. But they don't all have to be the same type of mutation. So for example, here you can see a map across this particular gene, CFTR gene, all of the exons, there are 24, and each of these symbols represents a type of mutation that results in cystic fibrosis. Notice that there are missense, nonsense, frame shifts, deletions, and splicing mutations that all result in the same disease. However, these often have differences in age of onset or how bad the disease is or how well they respond to treatment. So even if you have two patients that have the same disorder and a mutation in the same gene, there's no guarantee that their treatments will need to be the same either. So it's important to keep in mind that all of these different things we've talked about today can contribute to the same thing, and that makes learning about those things quite complicated. So that's all I want to talk about for mutations today. We've discussed the different types of mutations, different ways that mutations can be caused, and different ways that mutations can be repaired. We've also discussed the idea that mutations do not have to occur within the coding region of a gene for them to have negative effects on the gene itself, either because they affect the way that it's expressed or the way that it can be used.